Hi class, Professor Nigel McMillan here, and sorry I couldn't be here today for this lecture. Unfortunately, due to some conference travel and some timetabling clashes, I've had to pre-record these lectures. But as I said in my note, you can email me if you have any questions about this or make an appointment. If we have a lot of questions from the class, we can also organise a Teams meeting. So for Future Therapies 1, we covered gene therapies, genetic therapies, CRISPR, siRNA, and the like. It was really the future for therapies for cancers, metabolic diseases, lots and lots of things, mental health disorders, etc. What we're going to talk about, or what we talked about, I guess, in that lecture was replacing or repairing genes that had gone wrong in cells. For Future Therapies 2, we're going to talk about stem cell therapies, and this is really more taking a sort of a whole cell engineering approach to future therapies, where we might have whole classes of cells that are now defective, for example, in something like diabetes, where we know the islet cells have been either destroyed or don't function anymore. In the case of type 1 or type 2 diabetes, when we have other issues around retinopathies where our eyesight may not be working well, and we have issues with our cone cells or whatever it may be. And so in this case, we're really looking at being able to replace a class of cells. And the question is, where do we get that class of cells? How do we go about making that class of cells? And this is where we're going to be looking at stem cell therapies. So hold on for about the next 30-40 minutes or so, and we'll go through some of the reasons, some of the ways we go about doing it, some of the major discoveries in the field, and some of the things that are going on near and far from home in terms of stem cell therapies, because believe it or not, Griffith is one of the world's leading stem cell therapy universities. So it's a great place to be if you're interested in that. So what are we going to learn? As I said, we're going to learn what stem cells are and how this fits in with our biology, and what it actually means to be able to find them, make them. What are the different types of stem cells? So we have a number of different sorts of stem cells, as depending on where we source them from, and what those stem cells are capable of. And then we're going to talk about what stem cell therapies are, what they could be used for, what they are used for, because there's actually a very famous type of stem cell therapy that we are undergoing many times a day all over the world, and that's essentially bone marrow transplantation. And what is it good for? What could we actually use it for? So, as usual with lectures, in this particular module in 3022, this lecture is your starting point, really, to give you a primer for what's going on in this field, from which you can then learn more yourself in your self-directed learning and in some of the other classes associated with the course. Okay, so stem cell therapy, what is it? What might we be thinking about using it for? The possibilities are quite endless, but ultimately stem cell therapies could be used for treating diseases, and in the case of bone marrow transplant, things like leukemia, where we have bone marrow stem cells that basically fill the hemopoietic niche. That is, they make our T cells and our B cells and our macrophages and our platelets, all from the same sort of stem cell. And when these things go wrong, one way to treat them, of course, is to completely destroy this stem cell niche and reimplant it with new stem cells. There's a lot of stem cell therapy looking at how we can alter the course of aging. So some of us are older than others, but can we take grey hair and make it black again? Or can we improve our eyesight or our muscle tone or our bone density? Are there the vagaries of aging? Can these be reversed by perhaps using stem cells? And of course, this is a search that has gone on since the beginning of man. Everyone has tried to avoid the inevitable consequences of aging and even death. And what might that be? The elixir of life, the philosopher's stone, whatever it may be. And of course, we can think about things like injuries, where we have very significant repair issues. Let's say we lose sight or hearing, or in the case of motor accidents or spinal cord injuries, where we lose mobility we lose use of our legs, or legs and arms in the case of quadriplegia. So how might we be able to fix that bridge where we've got a break in the spinal cord? And that, of course, is research that's going on right here at Griffith. Okay, this particular YouTube video is a really useful one to explain stem cells for you. I could have it played during this video, but I've given you the link for you to go and watch it at your own leisure. It's just a bit of an issue around playing these YouTube videos in this particular setting. So it's about 6 minutes long, and it's well worth a watch. So go over and have a watch of that now, and pause this, and then come back and watch it. Watch the rest of this lecture. Okay, so we'll just move on to the next slide. Okay, so what are stem cells in its sort of most basic setting? Well, as you can tell from the name, obviously, you know, these are cells that actually are the beginning of all cells, right? They contain the ability to become many types of cells, and these are ultimately self-renewing cells. That is, they go on pretty much forever. Now, the only things that usually go on forever in the human body are cancer cells, which obviously are not normal, and stem cells, and of course they don't really go on forever. They also have a lifespan. Now, normal cells, when we think about them here in yellow on the left, divide by division, normal symmetrical division. So one cell becomes 2, 2 cells become 4, and 8, etc. And those cells normally have a set half-life, and this is usually set by a number of biological mechanisms to determine how long they will divide for and how long they will live, including the telomeres at the end of chromosomes, and every cell type has a different sort of telomere length, but essentially acts as a biological clock, at which point when the telomeres get too short, the cells know that it's time to die by some sort of apoptosis mechanism. And all normal cells are essentially birthed from stem cells and then die, and that's normally how it works. Think about our skin cells. Our entire skin is turned over every 3 weeks or so. Your house is full of dust. You want to know what that dust is? It's all skin, essentially, from you and all your animals and whatever else might live in your house. But for stem cells, this is quite different. They undergo what we call asymmetrical division, and that is, a stem cell can divide here on the right into a differentiated cell in the green, and in this case another stem cell, right? And this is asymmetric division. Now the green cell, which is a differentiated cell, is down the path of differentiation, may divide a number of more times, but it has a set length. Whereas the self-renewed stem cell in the yellow can now divide again asymmetrically. So we always have a stem cell and then populating a population, I guess, of differentiated cells. So this is the difference. Stem cells can self-renew in this sense, so make more copies of themselves in that way and be endless, so that we can, whereas a differentiated cell, eventually has a normal lifespan and dies. And so these give rise to a number of unique characteristics that stem cells have. They're unlimited, their ability to be unlimited in self-renewal. And of course, you know, leading people to realize that this is perhaps the secret to a longer life, or and the like. And what we know is that as people age, their stem cells start to become less efficient. We don't replace things in the same sorts of ways, and things don't function as well, leading people to the point, well, you know, if I inject myself with, you know, serum from young organisms, then, you know, they may take on younger properties. In fact, some animal data supports that. The factors that are in the bloodstream, which are a lot higher, can sort of renew old cells to a degree. So they have unlimited self-renewal, and then they have the ability to differentiate. So this depends, of course, on a number of things. And differentiation, or what sort of cell they become, depends on the signals they have, the external signals they're given, and also on the internal signals they're given, that is the genes that are actually expressed to tell them to do this. So a stem cell can differentiate, and they will maintain what we call tissue homeostasis. You know, our skin renews quite nicely, our blood is made, you know, etc., etc. But of course, then we go on two sides. If we do this improperly and we have mutations, this sort of control gets lost and we might form cancer. And the other issue is if it in the normal, as things goes on, as the system wears out, we start degenerating and we start aging. And so this is really the conundrum essentially of stem cells, is to making sure we get that balance right. And how do we actually get that balance right? And how do we manipulate the system to actually be medically advantageous, to repair broken systems and fix things, as we all hope to do with you know, medical interventions and therapies. So if we were sort of to define a stem cell, we would say that they can continuously divide and give rise to new copies of themselves. So that's that self-renewal. And then they can give rise to other specialized sorts of tissues or cells. And realizing that, for example, the ultimate stem cell, you know, is the egg, and the fertilized egg, because that egg divides and divides and divides and then gives rise to every sort of tissue, you know, in an organism. So that, you know, one egg then goes on to make the kidney cells and the lung cells and the bones and the muscles and the brains and the nerves of an organism. It's an incredibly beautiful biological dance, orchestrated by merely our genes. And how do we actually manipulate and control this has been, I guess, the point of a lot of study over a long time. So, of course, and also what decides whether a stem cell remains a stem cell or whether it becomes more functional. We do know that there are steps down the stem cell where we have specialized blood stem cells and skin stem cells, so they can start to semi-specialize rather than being completely potent and making everything, or totipotent as we call it. But ultimately, of course, the main function of these stem cells is to maintain our bodies, and that is to continuously replace cells that are worn out, to repair them, defective cell types, and regenerate normal cells. And of course, this is why we call it regenerative medicine, a really important area of biology. And if we were to think about classifying stem cells, as I said, there are really, it really is done according to potency, so that we have a number of different sorts of stem cells. So totipotent, which of course is something that means that they can make all cells, or pluripotent, they can make some sorts of cells, or multipotent or unipotent, etc. And then we also define stem cells in terms of where we get them from. Obviously, the most obvious source of stem cells are embryos, because that's what they are, stem cells. But we can get stem cells from adults. And one of the issues that is going to become apparent as we go along here is that sourcing embryonic stem cells has a number of ethical considerations that the field had been beset with and is now essentially solved through technology. But a number of people, groups, religions, scientists even objected to the use of embryos in some of this research. Okay, so if we actually were to sort of diagrammatically outline stem cells and where they're used, this is the sort of diagram you would probably come up with. So the ultimate stem cell is the fertilized egg, right? And that is a whole bunch of what we would call totipotent stem cells. Those cells, an 8 cell embryo, 16 cell embryo, every one of those cells can become, in fact, an entire organism. And this, of course, is where we get identical twins or triplets or quads. You know, this is where some of those cells break off, and yet they're genetically identical and form to become twins or triplets, etc. in the womb. They form into a blastocyst, and these blastocysts now start to endear and develop what we call pluripotent stem cells. So they're starting to specialize now at this stage, where we might get stem cells for bone, or stem cells for nerve, or stem cells for muscle, etc. And these isolated pluripotent stem cells, if you can get them from the inner mass, can be cultured. So if you hear people talk about pluripotent stem cells, pluripotent embryonic stem cells, that's what they're talking about. That's the cells which they've got from inside the blastocyst. But ultimately, as it goes on, we get hemopoietic stem cells, neural, etc., mesenchymal stem cells, which are the ones that make the connective tissue, the bone, etc., and blood. So when you hear me talking about these terms as we go along, this is what you can use to go back and refresh your understanding of what I mean by each of these terms. So totipotent, right? These cells can form an embryo or a placenta. Pluripotent, right? Which are the more specialized stem cells, right? This cell can just form the embryo at all, right? Multipotent stem cells, and these cells, as I say, specialized and more specialized, and they can't make an embryo, or they can't a whole embryo, and they can't make an embryo and a placenta. Right, and so if we look at the history of stem cells now, we can actually see that the term itself was developed about 120 years ago or so by a Russian histologist, and he coined the term stem cells when studying blood. Blood being relatively easy to get out and study, so it's always been useful. Excuse me. But we really couldn't grow the first human cells in culture until the 50s and 60s. And that was when we started culturing HeLa cells, and these are cells that came from Helen Lane, a woman who had cervical cancer in Baltimore in the US. And cancer cells, of course, grow because they're immortal, right? These can divide in an unlimited way, And that's because they've forgotten how to die, and also they've been mutated so that they now have continual growth signals. So these are immortal cells, but not in a good way, in a very bad way. And of course, the flip side of these stem cells is unlimited division is cancer. We really started getting an idea more about the sort of cells that made up our blood in the study in the 60s of hemopoietic stem cells, and started to realize that leukemia as a cancer was a disease that had a relationship with stem cells in the blood. In the 80s then, another leap of 20 years, and we started to study embryos and mouse embryos. And this was a Nobel Prize in 2007 for work done in the 80s, where the first embryonic stem cells were derived from mouse embryos. And in 1988, umbilical cord, and then of course in 1998, the first stem cells derived from humans. This was, I guess, a gold moment. The study went on in 2006 and 2007 when we started to be able to get inducing pluripotent stem cells from somatic cells. Now, what do I mean by that? Well, this is now taking those cells that are differentiated and dividing, or will eventually stop dividing, and reversing the course back to pluripotency. And this was done by Yamanaka and Takashaki. And this was also a Nobel Prize, by the way. And discovering the factors in 2012 by Yamanaka and Gurdon about what factors would take mature cells and reprogram them to become pluripotent. And then we have some stem cell banks that started in treatment. So we've really gone on quite a lot. As I said, when we think about the sorts of classification of stem cells, you can source them from IVF embryos or aborted embryos or cloned embryos. And the sorts of stem cells that you can get, adult stem cells, bone marrow, placental cord, and mesenchymal stem cells is the adult. So in the embryonic we have those. These are pluripotent and you get a large number. But one of the ethical concerns, or there are ethical concerns, but they also may cause immune rejection, because of course these are not your own stem cells. The advantage of adult stem cells, which we can get from bone marrow, as I said, placental cord blood, which is why some people save it, or mesenchymal stem cells. These are multipotent rather than pluripotent. You can get limited numbers and they're difficult to isolate, but there will be no immune rejection. That is, they're syngeneic, they are you. And we have very little ethical concerns. So when we think about the very first, I guess, iPS cells, these pluripotent stem cells, was when they were reported to be able to stimulate an adult stem cells into a pluripotent state, and these were called induced pluripotent stem cells. And there were no embryos, so this eliminated all the ethical issues. And there was this paper here, and you can see in the Cell in 2006. And these cells could be taken from the same person, so you didn't get immune rejection. So it really fixed a lot of the problems that surrounded embryonic stem cells. And these became known as the Yamanaka factors. Right, and I'll tell you what they are in a minute, but it turns out there are 4 things that you need to give to a cell to reprogram it into pluripotency. And this went along with research in the 60s by an Englishman, John Galton, who removed the nucleus from fertilized eggs from a frog and replaced it with the nucleus of a cell taken from a tadpole's intestine, and showed that in fact you could grow a new frog. So these 2 important steps, even though they were many years apart, led to the Nobel Prize. So what are the Yamanaka factors? The Yamanaka factors are essentially factors to reprogram, and it turns out that there are 4 of them. They have very strange names, but they're essentially transcription factors. That are things that are proteins that cause genes to be transcribed from the genome. They're called OCT4, SOX2, KLF4, and CMYC, which you may remember from if you've done cancer work, is one of our normal but also genes that can be mutated to cause cancer, right? And so it's got a bit of an asterisk beside it there. There are other factors that could induce it, Lin28 and NANOG as they're called, which would help with certain, but ultimately they could show they could take these 4 factors, right, and they could take, say, a hemopoietic cell or a cell, and they could make them into neurons. They could reprogram them into pluripotency and then coax them into become cardiomyocytes or hepatocytes or whatever with other factors. And so we ended up with the idea is, well, how do cells know what to become? So we had to then study, well, how do we make, if we reprogram something into a pluripotent stem cell, what do we then expose it to become different sorts of cells? So genetic engineering, essentially, to help them become the sorts of tissue that we wanted, right? How does an eye cell become a nose cell, etc.? So here's essentially the genetic engineering reprogramming system. So adult fibroblasts, they're very easy to get, were then introduced using usually viruses for gene therapy. These factors, KLF4, SOX2, MYC, NANOG, OCT34, and Lin28. And this would reprogram them back to pluripotency, so send them right back to the beginning. And from these iPS stem cells then, as I say, you could then show that you can make all these other factors. And this issue of recreating pluripotency was the big secret to the whole thing as to how it goes. But each one of these systems, whether you take those iPSs and make them pancreatic cells or neural cells, required exposure to other factors to encourage them along that pathway. And this is essentially how it's done, right? You ultimately can reprogram them with these factors, and this is done with lentiviruses. This is just a diagram about how we make lentiviruses and how they're made, but for the purposes of our lectures, it's probably not really important. There's some notes here with each of these slides if you want to have a look at them. Okay, so what are the pros and cons of iPS cell technology, right? Because there are certainly some. Well, the pros are the cells can be genetically identical to a patient, or a donor. So, you don't get any immune rejection. Really, ultimately, this is really important. You don't need an embryo. As I say, that removes a whole lot of ethical dilemmas from the whole system. But they're not without their problems. The problems might be that the cells might still have a genetic defect. So, if you are trying to treat a particular disease from a patient, say cystic fibrosis, well, every cell in that person's body will actually have the mutation if they were born with that contains that particular issue, or Huntington's or whatever it may be. So you haven't repaired the gene necessarily, you just made more of the cells that you might need, right? One of the other concerns was that some of the genes used, or one of the genes used to make pluripotency work, that is c-Myc, is a cancer-causing gene. If you overexpress c-Myc, or it's mutated to become on all the time, that is a pathway down the road towards cancer. So that can be a problem. And then the viruses that we use to insert these genes are lentiviruses. They might insert genes in places we don't want them. And that was a concern because we'd had some gene therapy trials in the 90s and 2000s, particularly around young boys in France who were on a trial for severe immunodeficiencies, SCID as we call them, where they got treated with a lentivirus to replace the gene that was missing, and several of them got leukemia, and it turns out it had inserted into a particular place in their genome that turned on a gene like c-Myc to cause this. Ironically, interestingly, it was only in the French trial this happened. It didn't happen in the English trials or the American trials for this disease. So there's something peculiar, but nevertheless it introduced a worry. But obviously for now these cells are perfect for making and studying diseases in the dish to promote basic research. But we've obviously got a lot of details to work out. Now, what are factors known to affect stem cells? How could you make your own stem cells work better? Because you've all got them, we've all got them. Maybe it's just a matter of making them really healthy. Well, it turns out the things that make stem cells work are the things that actually make people healthy. So that's probably of no surprise. Low stress levels, regular exercise, mental stimulation, learning, a use it or lose it sort of attitude, I guess, to muscles and neurons and the like. Healthy diets, avoid excessive drinking. So it's no surprise that, in fact, things that give you healthy lifestyles are in fact things that make your stem cells healthier and work better. Okay, now if we think about adult stem cells just in terms of therapies and where we're going with these, ultimately hemopoietic stem cells are interesting because they give rise to all types of blood. Mesenchymal stem cells are very interesting because they can give rise to things like osteocytes, chondrocytes, adipocytes, and other connective tissue. Neural stem cells will give rise to neurons and astrocytes and oligodendrocytes, epithelial cells, skin cells, and cord blood stem cells. Now, and this is often very much, we know we can freeze cord blood. I have a cord blood cell bank. It's part of our biobank here at Griffith. And these will contain many, many stem cells for those particular individuals for which that cord blood came from. And so parents who might have a history of a genetic disease in their family will choose to have their baby's cord blood frozen down so that if they need a transplant later on, they actually already have this available. It's not for everyone. And you probably wouldn't just do it just on the off chance, more likely if there was a history. Now, if we think about the therapy, in 1968 the first adult stem cells were used for a bone marrow transplant. And in fact, this is our most common stem cell therapy. This is why we mentioned blood first, I guess. And so there were two big, well, there are a number of big tricks to it that we learned along the way. The first was that for patients who had needed a bone marrow transplant, normally because they had some sort of leukemia that had not responded to therapy, and we've come a long way in chemotherapies and radiation therapies and cancer therapies for leukemias. The big one, of course, now being CAR T-cell therapies. But ultimately, we knew that we firstly had to get rid of the current stem cells because they were obviously defective. And that is, we had to get rid of them out of the bone marrow. So what often happens, or what now happens with bone marrow transplantation is you will get drugs to mobilize your stem cells more out of the bone marrow, and then there'll be radiation therapy to kill them. So that we kill off everything, and it's like clearing all the weeds out of the garden so that you have a nice new garden bed where your new stem cells can plant themselves and grow. And so this leaves room for them. But this is and then we harvest from a donor stem cells, and then we inject them into patients. This is a very routine thing now, in a sense. Bone marrow transplantations become very common. Still a few issues around bone marrow transplantation. The first is whether the graft accepts, so we get engraftment. And secondly, if we don't get sometimes graft versus host disease, that is, the new bone marrow stem cells actually perceive their new home as not themselves, and so of course this is why we have immune suppressive drugs that have to be taken. So it's not the answer to everything. There are many potential uses of stem cells for sure. Traumatic brain injury, stroke, Alzheimer's, any of those neurological conditions. Spinal cord injury, obviously. Myocardial infarction or heart disease, can we replace the stem cells of the heart to make sure it self-repairs? Immune disorders like diabetes or Crohn's disease. Can we repair muscular dystrophy? All sorts of things. Rheumatoid arthritis, many things. So here's just a procedure that we had for bone marrow transplantation. So here we're going to infuse healthy blood forming stem cells into the body, right? So we stage one, we receive a pre-treatment to release all the hematopoietic stem cells from the bone marrow. We collect those from the donor or the patient. They're separated and removed. Stem cells are frozen. They can be put back. But normally we have them from another donor. Right? Well, there's two sorts. You can have an allogeneic, which is donated stem cells from another donor, or autologous, which are your own stem cells. Now, once again, here's a really good video for you to go and watch, copy and paste, and watch that. Pause this, go and watch it, and come back. Okay, so just for the last couple of slides, we're just going to look at some of the sort of interesting recent trials around stem cell therapies. Probably could have included CAR T cells for this, but that's just a new form of leukemia. But that's more about genetic reprogramming than stem cells per se. Okay, so I say the latest trials. I like this one because it's from 2023, so it's 3 years old now. And it's from the University of Cambridge, looking at multiple sclerosis. So MS obviously is a disease where your own body's attacking its the myelination around nerve cells, and we lose control of nerve induction and therefore control of our muscles. Now, this was a phase 1 trial. So remember, clinical trials happen in phases. There's a whole lot of stuff that goes on before things get to humans in terms of toxicity testing and animal trials and things. And phase 1 is the first human trial, usually with a very small number of patients, and it is merely to ask the question is, what's the safe dose? Can we give something to people and it's not going to make them sick or worse? And can we dose that up? So normally we start with one patient at a time, give them a dose, and then we move on. So usually a very small number of patients, in this case 15, and ask the question. If it looks like it passes phase 1, it's kind of safe, then we can go to phase 2, which is really upping to find the mean, the best tolerated dose, what's the highest tolerated dose for something, longer treatments, etc. And phase 3 is really, once that's all worked out, we can do a big trial and ask the question, does it work? So the phase 1 trials aren't really designed to show if a therapy works, but sometimes you can get a clue from the small numbers of the first phase 1 trial. So this is 15 patients, and these were embryonic stem cells that had come from a miscarriage. And what they were looking at here was a study design where they had 15 patients, average age 50. They had a mobility score they were looking for of what they call an EDSS, around 7.6, and disease duration for this cohort was 14 to 30 years. And what they were looking to do was they harvested some neural stem cells from a miscarried human fetal brain, expanded these, made them multipotent, and then were injecting them into the brains of patients with MS in this case. And so what they found was, and then they were doing it in increasing doses, and essentially they did all these studies as they went along, follow-ups at 1 month, 3 months, 6 months, 9 months, with MRIs, looking at spinal fluid and serum, and looking at their mobility scores and things like that. The upshot was, and here's the paper you can get from this link here, was that there was no worsening of symptoms over 12 months following treatment, which in itself is significant. There's another link here which you can look at. And it's just some of the data from this, but ultimately looking at, in this case, how many lesions they had, and you can see things sort of stabilized here, or normalized lesions slightly increased. You could look at the variation over time, and then you could look at the parenchymal brain volume changes over time, and actually went down. So this was in the course of this thing was a good thing. So this is looking like a promising therapy. Pretty involved though. If we think about what's happening here at Griffith, and I teased you that Griffith is one of the centres for this sort of therapy, and really led by one group here at the Clem Jones Centre, and led by Professor James St John, who you might get as a guest lecturer in this course, depending on he's a busy man these days. And this is work that was started by Alan Mackay-Sim in what was then the Ursitis Research Institute. He is the late Alan Mackay-Sim now, but James has carried on his legacy and developed in a really fabulous way this therapy, which was to repair spinal cord injuries using stem cell as a repair. And in fact, this trial has started this year. They're doing an actual clinical trial, so they've done a tremendous amount of preclinical work, and they're now doing a human trial. And the way that this trial works is that patients who have spinal cord injury do 3 months of priming rehab, and this trial is over a year, and then they have a surgery. So basically this is autologous, that is, it's their own stem cells, so they get a biopsy from the inside of their nose to obtain cells. Now why do we have stem cells in our nose? It turns out that our olfactory bulb in our nose, where we smell stuff, and as you can tell today I can't really smell very much from my cold, but that's the joys of travel. It turns out your nose, the nerve endings in your nose, actually are the only nerves in our bodies that actually replace themselves every few days. So they're actually growing nerve cells, and there are some stem cells in there. So they can be easily gotten to with a little biopsy up your nose. And we can get these stem cells, or James and his team can get these stem cells. They can expand them in this three-dimensional bioengineered nerve bridge. And this is a bridge that's going to extend over the injury of the spinal cord. And that's going to be transplanted into patients at the injured site. And they undergo 8 months of regenerative rehabilitation, and then they'll assess whether it's worked or not. Their work in animals is quite remarkable. and there's every chance that this will work quite well. Okay, so what did you learn hopefully in this sort of primer for stem cell therapies? Because we could spend a whole course just talking about stem cell therapies, but we don't have time for that. You've learned what stem cells are, you've learned what the different types are, you know what pluripotent, totipotent is, you know what induced pluripotent stem cells are, where they come from, what we use them for, you know what autologous means versus allogeneic, right? And you've learned a little bit about what stem cell therapies are and what it's good for. If you have any questions, as I said, please do not hesitate to email me. My email's public in the university directory, or just Google me. Otherwise, knock on my door in 305 3.31 or call me if you need to, and we can sort anything out. Thanks very much, and hope you have a great rest of this course.